biological experiments, and it has many usage and applications in like a diverse area, which also includes the research project that we're doing right now in our lab, which I'll talk about later. So let's start off with introduction of what flow cytometry is. And here we have definition from Thermo Fisher, which some of you might know is a really famous company for making biological equipments. And so the main focus of the definition is that it's a cell analysis technique as using cells and a flowing fluid stream. The way how it memorizes function is simply by its name. As you can see, if you divide the name by two parts, the first part, flow, can refer to the flowing stream, whereas the second part, cytometry, can simply refer to cyto, which is a loan word borrowed from Greek, meaning cells, and metry, meaning the process of measuring. So combining all of that, you simply have the process of measuring cells in the flowing stream. And the right side, we simply have a really basic demonstration of the setup of a flow cytometer. And from the top, we have sample cells coming down all the way to what we call the interrogation point, where the laser light source hits the cell and detects it. And the cells are also forced down by something we call a sheath fluid, which allows them to pass in a single cellular manner. Moving on, we'll talk about how a flow cytometer detects cells. It mainly does that in two ways. The first way we call forward scattering. It's simply when a light path, in our case, it's a laser beam. It hits a cell, and the cell would cause forward scattering depending on the size. Now, the extent of the forward scattering is directly proportional to the cell size, and therefore the larger the forward scattering means that we have a larger cell. And then we have a detector that's directly opposite to a laser beam. The second way of detecting cell is by size scattering. And size scattering is directly proportional to the granularity in the cell. And the granularity simply means how complex a cell is. So for example, a cell has a nucleus and other intercellular compartments, which are really dense. So therefore, when the light hits it, it's not going to pass through, it's going to bounce back, and therefore we have the size scattering detector at the side. So in here, I've prepared a few cell models for demonstrating forward scattering and size scattering. I really appreciate it if we can turn off the lights at the front for a while, like the front lights. Yeah, thank you. So as you can see, this is a smaller cell, and this is a cell of a larger size. And so when I shine a laser beam here, this is exactly how it's set up in a flow cytometer. When a cell passes through, it will cause scattering of the light. And it might not be seemingly obvious, but a larger cell would cause more scatter of the forward light. Because if you think about it, logically, a larger cell would obviously take more time to pass through at least the interrogation point, and therefore cause a more forward scattering. And in here, we have a cell that demonstrates side scattering. As you can see, the first one is a really simple cell with basically no intracellular compartments, whereas the second one is more complex. So again, when I shine a laser beam through it, the first one can pass through quite well, and therefore less light is scattered 90 degrees particularly or backwards. Whereas because the second cell is more complex, when I shine light through it, it doesn't go through, it reflects backwards. And therefore we have the side detector at the sides instead of opposite of the light source. Now, so we have another diagram demonstrating of the setup of a flow cytometer. You don't have to know the details. What you need to know is that we have a light source here. In our case, we don't necessarily only have one laser beam. We usually have 2 to 3 laser beams that's of different wavelength that shines and hits the cell here. And again, FSC simply means forward scattering. So this is a detector of for the forward scattering. And here we have the detector for side scattering. And by the way, we have a really quick pop-up quiz. So I really appreciate it if you guys can go to this website here and enter the code using your cell phones. That's not a joke, I mean it. Yes.
Yeah, just in the meantime, large cells and small cells, they flow the same rate, right?
Excuse me?
They flow the same rate through the detector. So you ensure that they flow the same rate.
Yes, because you control the pressure of the flowing stream.
So the concentration is fairly low of the actual cells.
Well, it depends on how quickly you take them up. You can actually control the pressure, and they have these, well, the sheath fluid that I was talking about just now. They have a technique called hydrodynamic focusing that ensures the cell to pass through in a single cellular manner.
Okay.
I'll refresh it pretty quickly here. Yep. You have it?
Yes.
Okay. Oh, by the way, so these are the three cell types that we're going to deal with. And I simply set some arbitrary values for the size and granularity. As you might have remembered, granularity simply means the complexity of the cell. And yes, you do have 4 options, so it's not that straightforward.
Sure, it's the same question.
Yes, basophils. Oh, sorry, I meant neutrophil. Yep. Yep. For the first question, it's actually neutrophil instead of basophil. Sorry about that.
So what is SSC and FSC?
Oh yes, SSC is size scattering and FSC is forward scattering. So as I wrote here, so forward scattering tells us about the size, and size scattering tells us about the complexity, which I've gave in the arbitrary values that I've made.
What is the cell type in the PowerPoint? The question you asked.
Yeah. It's neutrophil instead of basophil for the first one.
Okay.
Yeah.
5.
Oh, you did that wrong one. Because I'm not seeing anything here. Oh.
Submit.
Okay, so the first question is about the neutrophil, and quite a lot of you got it right. So the neutrophil would be D, because it has a size of 5, which is like the highest value. So right here, and the granularity is also the highest. The granularity meaning the complexity, which is the size scattering here. So matching 5 and 5, we have a population of D here. So let's move on to the second question. And yes, most of you got it right as well. For the second question, the lymph site would be B right here because it has a very small granularity and the size is quite small as well. It's 2 comparing to the other ones which has a size of 5. And moving on to the third question. This is the last question, by the way. if you go to right first time, it's that's a C. Because the size is 5 and gravity is 1.5. So we have an extra value of A here, and I'm going to explain what it is because yeah, this is the answer that I found in the internet as a reference. The lymphocyte would be here. They're very small cells and with quite low complexity, and therefore the forward scattering is here, meaning that's quite small compared to these two cells that we have here, and that it has a low complexity shown by the size scattering, whereas neutrophils is most complex, therefore, and it's very similar in size compared to monocytes, therefore it's right there. And A is simply the cell debris that we might have in the sample solution when the flow cytometer sucks the cell up. There might be some dead cell, that cell that's lysed. And the reason why I put A there is not just to trick you, but to show that most of the time our data is not as ideal as what we would wish it to look like. We always have some background here and there, and we have some cells that we might not know what it is, and it's really important to know that where our cells might be. Yep, moving on, we're going to talk about how flow cytometer detects sub-cell types. So even within these regions of lymphocytes, we still have, let's say, T helper cells, T helper lymphocytes, and T cytotoxic T lymphocytes, which are the same size and the same granularity. However, they have completely different functions. So how do we differentiate against them? And we do that by a phenomenon known as efflorescence. And I'll talk about it very briefly here. Basically, it's when we have a light source, in our case, it's a laser beam, that shines to something we call a fluorophore. A fluorophore is simply a molecule that's able to undergo the fluorescence phenomenon. So when light hits the molecule, it excites the electron to a higher energy state. And while it drops back to the ground state in a nanosecond scale, it emits fluorescence light that we are able to detect. So in order to detect between cells that are the same size, but they have the different functions, we use something we call antibodies, which recognize very specific cell surface markers. So even if these cells are the same in size and granularity, they still have stuff that's expressed on the cell surface that is very different, and which gives us an idea of which cell type it is. And so we have these antibodies, which this is the variable region, so it's very specific and recognizes only specific and unique cell surface markers. And because we have the fluorophore attached to it, when we shine laser through it, it does emit a fluorescence signal which we can detect. And here, the large circle is simply a cell, and the smaller green circles and the orange one right here is simply indicating that it's a cell surface marker. And this diagram is to demonstrate how antibodies with fluorophore right here attached can attach and recognize these cell surface markers. Yeah. And basically when a laser beam shines through it and hits it, we do receive fluorescence emitted from the cells, and therefore it can hit the detectors. This is a simple diagram demonstrating the fluorescence peaks of a molecule. So this is the excitation peak, and this is something we call an emission peak. Notice how the wavelength of the excitation peak is always smaller than the wavelength of the emission peak, and that's because wavelength is inversely proportional to the energy, and the excitation would have like always a higher energy than the emission, because energy would always dissipate. Yes.
In practice, do you use one certain type of antibodies if you expect one certain type of cell, or do you use a mixture of antibodies, and depending on the wavelength you get back, you know which type of cell it is?
We do have like a live example later on, but usually we use like more than one antibodies to confirm. Yeah.
I'm curious, how do you do the cell sorting afterwards? Do you have a slide for that?
For cell sorting?
Yeah, after this, you want to sort labeled and unlabeled cells. You just do it manually or take cells, or there's automatic?
Well,
there's a charging
software
method.
to analyze.
You charge the droplets.
yes, there's.
So it's on the
procedures.
fly. So it's on the fly. So if it's fluorescence,
Mm-hmm.
then we sort it somewhere.
Yes, we do have that. It's not, I didn't include that part in my slide because for us, we can do that with a software that tells us which fluorescence we are detecting from the cell, and we can separate the data accordingly. But yes, it can sort the cell as well. Does that by charge, for example, is something that we'll talk about later. Some of the fluorescence, the molecule has like a charge, and they have these charge plates downstream of the laser point, the interrogation point, and which separates them by positive and negative charge according to the fluorescence molecule and the antibodies that's attached to the cells.
But what if you want to use intrinsic fluorescence, not the antibody? So, for example, if you want to,
you can sort the cells in that case. You can't see the signal, but
I saw a piece of work from Nancy's group. They're doing this flow cytometry on this amyloid fiber. So they have this some cells with amyloid fiber, A beta fiber. Some cells doesn't have A beta fiber.
Right.
The other fluorescence dye, something similar with a Congorat, but it's a fluorescence dye. Then you just use this technique to sort the cells.
okay.
With and without signal. I'm just wondering. When I was
They do use fluorescence. You mean, where are the fluorescence dyes? Is it in the cell or
Yeah,
do they
yeah.
it's in the cell?
No, it's segregated. So I think it's probably
Then
not
how they
inside.
attach it? I assume they kind of use the antibodies to
Flow
attach it.
together. Or you think it's also use antibody?
Yes.
Oh,
It's
so
a
this
fluorescence.
dye is attached to antibody.
Yes,
Not directly
yeah.
in the flow. Oh, okay.
Yeah, we have to incubate them before we apply them through flow cytometry.
No, but the antibody is here. You
Mhm.
actually target specific surface receptor. Right?
Yes,
So
we
in
do.
that case, it's really the dye will bound to the beta sheet fibril structure of the A beta, and that will separate the cells, subtype of cells. I think it's slightly different.
I think there is a method with, you sort them by droplets, right? So the cells come out of the machine like in droplets.
Yeah.
And there's a method for charging these droplets. And then
Mhm.
depending on the fluorescence, you can sort them by alternating the electric field at the end, which
Yeah,
direction
right.
the droplet will go. And that way you'll get cell sorting.
Yeah.
And you can sort them on the GFP or whatever, because you don't have to incubate with antibodies. Isn't that right? Can you sort cells which express the GFP?
Right, that's probably the
I think
I'm not-
you have slides later on talking about sorting.
Yeah.
Separate the tank. Yeah.
Do you have slides for
the
sorting?
neutral one, the positive charge, negative charge one? I see. I remember I saw it. You were preparing your slides.
Okay.
But then if you do the detection, and so what? I mean, after you do the detection, so what? It's not useful.
We can know the population of cells, the proportion of the population of cells expressing the specific cell surface markers.
Okay.
Because, for example, the project that I've done during my summer school in Imperial is that for T cells, when they activate, they have different cell surface molecule expressed compared to when they're not activated. And in that case, we don't have to sort the cells. We just need to know the proportion of the population of cells that express that signal.
Yeah, well, excuse me. For the further sorting about the cells, I mean, in my undergraduate study, I remember that maybe use a tool to detect the overall mass of the nucleotide or something.
The mass of the nucleotide.
Yeah, in the cells and to sort the cells into.
But how would that solve different cell types?
For example, maybe the sexual cells, which contains Y chromosome, another contains
You
X.
mean less nucleotide than somatic cell? Perhaps.
Yeah, there's many different ways you can do the cell sorting.
Right.
One
Yes.
question. Does the fluorescence need to be extracellular or intracellular or both? I mean, can they do, of course, extracellular is okay. How about intracellular? Like inside the cell, the fluorescence is inside the cell. Can it be sorted?
I think the method that Kiryu mentioned just now might be able to sort a cell by giving them a charge after detecting what if they have for example, the green fluorescence protein.
I mean, the light source is in the cell, not on the surface of the cell.
I think, yeah, as long as you can detect it, you should be able to sort it according to the electronic the charge plates.
But like inside the cell they have different components. If the light source is in the cell,
Yes.
how do you know whether the light emitted is because you mentioned about scattering by the organelle. Right? Is there any method they can correct this before they sort the cell?
The what?
If the light source is in the cell, you mentioned that the component will scatter the light,
Yes.
and it's
But
they
for
didn't.
fluorescence, we're differentiating them by color.
So, but you don't care about the intensity or. I mean, actually, they are using the side detector to detect the light.
No. It's different fluorescence detector, which I will talk about in the next few slides. They don't use the side detector for detecting fluorescence light. No, they don't.
Just intensity.
Yeah, just side scattering itself. They have different other detectors for detecting fluorescence light.
Maybe we should move on.
Okay. Yes, so after applying fluorescence, we need two types of band filters to separate the fluorescence signal, because otherwise, as demonstrated in this slide, it's a continuous signal. If we apply two to the same cell, we have like a really huge background and kind of purposeless, as we cannot really differentiate between the fluorophores. So we differentiate them by using bandpass filters and long-pass filters. Bandpass filter simply means that when light source hits a bandpass filter, only a very limited amount of the wavelength of super wavelength, which is in our case 630 nanometer bandpass filter, will only allow light from the wavelength of 620 to 640 nanometers to pass through. Whereas in a long pass filter, as its name suggests, only light of wavelength longer than 520 nanometer in this case can pass through, whereas all the other lights that have short wavelength will be reflected. And yes, it's essential for us to differentiate between different fluorophores. And here we have the setup of inside of flow cytometry, flow cytometer. And basically in here demonstrates two different types of the fluorescent detector. One is called the trigon detector array, whereas the second one is called the octagon detector array. We're mainly focused on the octagon detector array for the purpose of our presentation, as I believe it's more capable of demonstrating how it separates the fluorescent to the fluorophores. Here's simply the light electromagnetic spectrum of the visible region, just to remind you of the wavelength. And in here, we have, first of all, let's say when a light beam comes in, if it's it will first hit, if the fluorescence comes in, then it will first hit the 735 long pass signal. So if it's longer than 735 nanometer, let's say if it's around here, it's able to be detected by the detector here, whereas if it's shorter than that, it will simply be reflected until it hits its designated detector. So let's take an example of this green fluorescence here, which is around 530 nanometers. So when the signal comes in through here, it reaches the first 735 long pass filter, and as I explained, it will not go through the filter. Instead, it will reflect to the second one, which is 655. And since it has a wavelength of 530, it can still not pass through. So it will reflect until here, until here. We have a 502 long pass filter, and because our wavelength here is around 530, this time it can pass through, and it will be detected by our 530 the bandpass detector here. And these are just this diagram is to show that for the excitation peak, it's okay to use fluorescence of very similar excitation peak. In fact, sometimes we do that on purpose because we want to excite both of them for using the same laser beam. However, if we want to differentiate between different fluorophores or different cell surface molecules, we wouldn't, in an experiment, for example, we wouldn't use these three in the same experiment because as you can see, the emission peak is very close together, and therefore it doesn't achieve our purpose of separating and differentiating between different fluorophores. Instead, we would typically use the green one, the red one, and the brown one right here because they're quite separate. Yep, this is basically a summary of what I just said. As the laser beam shines and detects the cell, at the same time it excites the fluorophores that's attached to the cell by antibodies, and it will emit fluorescence accordingly. So the fluorescence signal A can pass through this long pass filter because it's longer than the wavelength and detected by detector A, whereas the fluorescence B has a short wavelength and cannot pass through, and therefore it's detected by another separate detector. Yeah, and moving on, we're going to talk about the application of flow cytometry. The conventional applications include cell counting and sorting, and as well as biomarker detection. Oh, this was a background we're talking about. Yeah. So they come out, the cells, after they are integrated by a laser, they come out as droplets, and they have these deflection plates which they can sort the droplets by giving them a charge. Oh, so this is the part which I mentioned is kind of related to our research project in our lab. It's from the paper that I read in the current protocols in cytometry. So it basically used flow cytometric methods to quantify protein-protein interactions. I've basically summarized what they did to set up for a flow cytometry experiment. So first of all, they want to look at something they call the RGS biotin. So they would first immobilize its first target protein by sticking it on the microsphere, which is also known as the beads. And the beads has like a pretty similar size to the cell, and therefore they can just use the sample to flow through this flow cytometer. And the second step would be to incubate the second target protein with a fluorophore. And the second target protein is here, it's G alpha O, and the fluorophore is called AF532. And basically, before doing this, they would first incubate the first protein with a bead, and then they would try to incubate the second protein with the complex of the first protein and the beads to see if they can interact and to see if they can bind. And the first step, which will be using additional samples such as inhibitors and competitor proteins to see that if they have stronger affinity than to the first protein, in that case, it would displace the second protein and bind to it. Whereas if it has a weaker affinity than the second target protein will still stick to the first target protein. And after that, they would use these samples and run them through a flow cytometer, and they can quantify the affinity by the populations of beads with or without the AF532 signals. So in this case, if it does have the AF532 signals, that kind of tells us that the second protein is binding to the first protein, which is also binding to the beads. Whereas if it detects a bead passing through, but it doesn't have the signal, it kind of suggests that it's displaced. The second target protein is displaced by other inhibitors or competitor proteins, or that it doesn't really interact with the first protein or binds to it. And this is part of the it's kind of a method that our experimental group came up with that can be used in our own research projects. So we can use flow cytometry as an alternative step to confirm the position of the influenza A virus polymerase after an in vitro transcriptional assay. So in here we have like a cRNA template with what I named fluorescence B attached to the end, and this template is attached to a bead. And so during transcription, the influenza A polymerase would first attach here and transcribe along. So after we do like an in vitro transcriptional assay, sometimes we see like a band with certain sites, but we're not sure if it's really the thing that we want. So we can use flow cytometry to confirm. So for example, if the influenza A polymerase with fluorophore A does successfully transcribe the whole template and goes to the end, there's a phenomenon, there's something called threat that will happen. So it's basically that these two fluorophores, fluorophore A and B, are specifically designed so that fluorophore A has an excitation peak as demonstrated here, and emission peak here. And the excitation peak of the fluorophore B would be very similar to the position of the emission peak of fluorophore A, and has a separate emission peak which is very different from the emission peak of fluorophore A. So something called the fluorescence resonance energy transfer would happen here. So if the two molecules are very close together, the emission, when they're excited by a laser beam, the first fluorophore would emit a fluorescent signal at this wavelength, which would excite the second fluorophore and hence emit a very different fluorescence comparing to being excited by itself. And this way we can kind of tell if the polymerase is very close to the end of the template or that it's not attached at all. Yep, I would like to
So why do you want to design this experiment? I don't know, what's the motivation of this? What can this experiment help?
For example, after immature transcriptional assay, sometimes we're not sure, we're looking at a few bands of certain size, maybe it's quite similar to what we want to see, maybe it's not, but we don't know what that product is, we don't exactly know. But after that, we can try to attempt to use a cRNA template and bind it to the bead, and we can run the, maybe at the same time of doing the immature transcriptional assay, we can allow the polymerase to transcribe along and see if they really did transcribe the entire product for the immature transcriptional assay. It's just like a basic idea taken from the flow cytometry method.
And calculate the kinetics of the enzyme. And there is a group that Li Zhe talked about last time that they are using this method to calculate the kinetics.
Don't quite understand. You only have a single probe.
Oh, we'll have like a lot of beads if we want to do a flow cytometry. A single one isn't going to give a signal for us to get.
So the kinetics you mentioned, you mean as function of time?
Yeah.
So you want to monitor the reaction as a function of time?
Yeah.
You only have a single.
control-
You only have a single label. How do you do that?
You can control the flow of the entire system, so that when the enzyme and the beads meets, and you control the time when they react, so that you can monitor the time.
Really?
Yeah. Just like the paper talked about.
No, these are talk about it's a stop flow, right? You're talking about stop flow?
I think the idea is the same.
Okay, so you want to use this as a stop flow.
So yeah, when the two reagent meets and you incubate some time to see how.
But then your detector is at the end, right? What if the reaction is halfway, then you won't be able to see anything?
We can add the B fluorophore at different positions to see.
Then how do you know which one? I'll combine with
Yeah.
Maybe we can add like different fluorophores.
Well, then you need to design a different fluorophore for different positions. You may use
We do have
tons
different
of
fluorophores, but we only need to make sure that it wouldn't interact with the transcriptional activity of the polymerase. That's something that we have to
So
consider.
you're talking about B, B1, B2, B3, they're all different. So
Yes.
now you only attach a B to the DNA RNA. Now you're talking about you're attaching different B1, B2, B3 into different position of safe RNA to monitor the
I
reaction.
mean different fluorophores. So fluorophore B, C, and D, for example.
Yeah, then you may need tons of the fluorophore to do that.
The template is quite short actually. It's a 14 or 15 something.
So this is how the stop flow guy do this experiment. I think they only have a single flow flow, right?
Yeah, theirs are very simple. They're just watching the red fluorophore and the green fluorophore, and how they have the resonance transfer.
Right. I think that's different from this. This is flow cytometry. So theirs is stop flow. It's different experiment. You don't need to. I mean, here my understanding is you're looking at this scattering, forward scattering, and
That's just to confirm the cell depletion.
yeah, yeah. That's why the flow cytometry is used, right? I mean, I agree. Experimental idea will be similar. In the stop flow, you also mix two things, right? Like here, you mix two things. You don't mix here. In the stop flow, you probably put the polymerase in the middle and the RNA on on the side to do a rapid mixing and monitor. But that will be much faster than flow cytometry.
Yeah.
So this will be at a millisecond time scale. It's totally different device. The flow cytometry, I don't think you can give such a high pressure. You need a very high pressure to push the system to flow at that fast to look at the millisecond dynamics.
Or maybe is there any way to like allow them to transcribe and stop at a certain point? And
No,
we
they
can
don't
control
stop. Basically the sort of the turn the spatial resolution to the temporal resolution we did in the micro. It's the same thing with microfluidics. You basically have two things mixed, and then on the two, a different position of the two is a different time point. You do the detection at a different spatial position of the two. That's how the stop flow works. So you're talking about stop flow, right?
Yeah.
I mean, this is nothing to do with. I don't think this device can do this.
Borrowing this idea to stop.
This is a right stop flow. You need a special pump. That's needs high pressure and the detector. I think they have this stop flow machine. Like it's a separate machine. I think in our department, Professor Guo probably has one stop flow. But that they use a UV detector.
With UV, you mean a UV excitation beam or?
UV absorption.
Oh.
The detector, I think.
We can use UV fluorophores to.
That's what they normally do in fluorescence could be a choice too. But why are we gonna do this? Why do we need to do this? Right, since we have a cryo-EM machine now, why not you just purify the complex and we get a structure out?
I mean, just an idea of how to calculate the kinetics of the enzyme, and how the two proteins interact with each other, and
stop flow, I think, is I don't know, may not be easy. We had a paper last year with microfluidics on the laser communication, but that is on the AIE molecules. The experimental side it seems not straightforward. You need to engineer these very tiny, teeny tubes with this high pressure pump to do that. If you want to do this, I would rather just do the rapid quenching gel. Maybe we should get a machine of that. Do you know how much is it, Peter?
No, that is at least half to 1 million.
Really? So expensive?
And very expensive.
The gel machine, quenching gel?
The quick quench, I think I checked one.
$500,000?
Yeah, minimum.
Okay, I didn't expect it so expensive. Okay.
Move on.
Yeah.
Okay. So moving on, I'll dedicate this part to the main research project that I did at the Imperial College of London last month, actually, during my summer course. So we use flow cytometry to detect different factors that can activate the T cells. So you don't have to know much about the T cells. You only need to know that it's part of our adaptive immune system, and that T cells need to be activated to kill pathogen and prevent us from getting sick. And activation of T cells can simply be understood as the stimulated proliferation of T cells. Yep. And here's the background required for understanding the activation process, but you don't have to you don't need to know all of these. You simply need to know that the T cell can be activated by two signals, signal A and signal B. And well, actually, these are the two signals that we use in attempt to activate the T cells. We're going to look at whether they can activate the T cells by their own or by combination of both. And so here's our experimental design using a 12-well plate as demonstrated here. And our design is that we have 3 repeats, what we call triplicates of double negative simply means that we only have T cells, inactivated T cells by themselves as a negative control. And signal A positive means that we have a combination of signal of T cells with stimulated by signal A in this column here. And signal B means that T cells stimulated by signal B for this column, and for the last column we have T cells with both A and B signals. Yep, and before showing you the results, this is activated T cells. So activated T cells will express surface molecules known as CD69 and CD25. You don't have to remember the names. You only need to know that it's very unique molecules that will only be expressed on activated T cells. And so we use antibodies to antibodies with different fluorophore, let's name them fluorophore P and S, to detect the presence of the CD69 and CD25 molecules. And as for a non-activated T cells, the antibodies won't attach to it because there's very little to know of the surface molecule expressed. And yeah, this is a summary of what I just said. Vectors A and B is tested for the ability to activate T cells, whereas we're detecting the activation of T cells by P and S signals, which is the fluorescent signal here and here. Yep, and here's our results. This is just a reminder of the previous design that we have for double negative, which is the negative control. We have like a baseline level of P and S, which we set as the baseline. And for the cells with signal A, it demonstrates that it has a higher P and S signal comparing to the baseline level, and therefore it suggests that it's activated. Whereas for the T cells with signal B, it has a very similar level, judging from the diagram, between this B and the double negative samples that we have here, which suggests that it has very little to no activation. Whereas the combination of A plus B also demonstrates a higher P and S signal comparing to the baseline, suggesting that these population of cells are activated. Here's our results presented in the table format. So we can see that the main focus of here is the UR region, because UR region means the upper right region, which is the region that we separated from above the baseline set by the negative control. And so, which simply means that if there are more cells in the UR region, that kind of tells us that there are more cells activated in the sample. And as you can see, double negative has very little amount of cells, cell population in this area, whereas signal A positive, so as T cells stimulated by signal A, has a much higher proportion of cells in this region, and this is the average of it. And signal B has, this is an anomaly, by the way. That's probably something that we did as an error in the experiment, which causes it to have such a high percentage, which shouldn't be the case. And this can be supported by the replicates that we have here, because these are very similar in value, which suggests that this is an anomaly, so we wouldn't look at this. And therefore, the mean for signal B is about 1.98%, and the proportion of cells for double positive is around 78% right here, and which kind of suggests that having both signal A and B have a synergistic effect. That means having a combination of them receives more level of activation, which is more than just a simple addition of A plus B. This is suggested because if we add up 70.2% plus 1.98%, it's still slightly less than 78% here, which does suggest that it's not simply an addition effect of signal A plus B, and it suggests that it might be a synergistic effect, although we can't say for sure because the levels are somewhat similar.
One question.
Yes.
Like because those two values deviate, right? But how can you know that the one with higher value is the abnormal one, not the two with the lower one?
Because we have 3 triplicate repeats, and having these repeats being so consistent kind of suggests that this is an anomaly.
But is there possible that you're doing for that 2 sample consistently wrong with some procedure? Because what I think is, you see that A plus B is higher than A only, right? Yes. But those contributions you suggest is by the combination effect of A and B. But why not? It's just because B indeed have some effect, just you set in the red part, and those two replicate are just maybe like.
If you add some even more higher than the A plus B,
Hmm?
if you add this, the average of this and 13%, it's even going even higher than this. And there is, like, we do have nodes doing an experiment, and some that suggest that we might have some area of error of transforming solutions when we're working with this well, whereas the other two wells, we don't have any records of possible errors for that.
Oh, so you did record that well have some.
It's possible.
Okay.
Yes.
Then it makes sense.
Yeah. And that would be the end of my presentation today. Thank you all for your attention.
Okay. So I think in the new semester, the group meeting will also be the same time slot. But maybe we can start at 2:15 rather than 2:30. 15 minutes after 2. So. Same room here.